So, good afternoon to all of you. So, in bioinformatics paper, in last lecture we have introduced about bioinformatics uh, and biological data and databases. Now, what are the different software or tools we are saying used in bioinformatics? So today we will discuss about tools used in bioinformatics. So, bioinformatics is an interdisciplinary field of science that combines diverse fields such as computer science, mathematical science, then statistics, and again it involves some engineering aspect, concept of physics, and many more in order to interpret and analyze biological data. So, the bioinformatics aims at understanding of biological phenomena. This is achieved by development of computational technique or tools which intensify the problem solving approach. So, with the help of computational tools किंवा कम्प्युटरच्या साह्यानं कम्प्युटरमधले जे सॉफ्टवेअर आहेत त्याच्या साह्यानं आपण हे फिजिक्स मॅथ स्टॅटिस्टिक किंवा इंजिनिअरिंगच्या साह्यानं किंवा इंजिनिअरिंग किंवा मॅथमधल्या जे काही फॉर्म्युलाज आहेत कन्सेप्ट आहेत त्याच्या मदतीनं कम्प्युटरद्वारे बायोलॉजिकल प्रॉब्लेम सॉल्व करण्याचा आपला एम आहे बायोइन्फॉर्मॅटिक्स. सो द टूल्स formulation in this field covers the data mining, pattern recognition, visualization, and machine learning algorithm which can facilitate the process of gene finding, sequence alignment, genome assembly, drug design, drug discovery, protein structure prediction, as well as protein structure alignment, prediction of protein-protein interaction, then gene expression, in addition to genome-wide associated study. So in this bioinformatics, we can visualize, we can have big amount of data, we can have a machine learning algorithms to study various processes in a biological system. that is एक सिक्वेन्सची दुसऱ्या सिक्वेन्ससोबत कंपॅरिझन. त्याच्यानंतर ड्रग डिझाईन करीत असताना सुद्धा आपल्याला बायोइन्फॉर्मॅटिक्सची खूप चांगल्या प्रकारे मदत घेता येईल. त्याच्यानंतर जीनोम फाइंडिंग, जीन एक्सप्रेशन, देन प्रोटीन स्ट्रक्चर. सो ॲज यू नो किंवा आपल्याला माहिती आहे की प्रोटीनचं जे टर्शरी किंवा क्वाटर्नरी स्ट्रक्चर फंक्शनल स्ट्रक्चर आहे, त्या स्ट्रक्चरमध्ये कुठले असे कोऑर्डिनेट्स आहेत, हायड्रोजन बॉन्डिंग आहे, अमिनो ॲसिड कंपोजिशन आहे, त्याच्यानंतर चार्जेस ऑन अमिनो ॲसिड आहे, त्याच्यानंतर दे आर ॲसिडिक ऑर बेसिक विच विल डिफाईन देअर फंक्शनल इन्व्हायरमेंट. सो दॅट ऑल स्टडी ऑफ प्रोटीन जीन्स then phylogeny sequence can be studied with the help of computer applying methods of mathematics, statistics, engineering, etc. So the we can say what types of tool किंवा असे कोणते कोणते टूल्स आहेत बायोइन्फॉर्मॅटिक्समध्ये ज्याच्याद्वारे आपण बायोलॉजिकल प्रॉब्लेम सॉल्व करू शकतो. सो फर्स्ट इज सिक्वेन्स अनालायसिस टूल. तर आता सिक्वेन्स इथं कुठली? द सिक्वेन्स इज आयदर ऑफ DNA or RNA or a amino acid sequence in protein. So suppose if you are considering DNA, then you have to take an nucleotide sequence, and if you are study at the protein level, so then you have to refer amino acid sequence. So sequence analysis tool, the sequence is a process of interpreting the information such as evolution, structure, and function from the sequence of nucleotides and proteins through the use of techniques such as similarity search, then search algorithms, conserve motif identification, domain annotation. So this step forms the basic for majority of studies, as there is a exponential increase in the number of sequence in biological databases due to increased innovation in sequencing technique. The following tools generally used for sequence analysis. सो सिक्वेन्स ॲनालिसिस बेसिकली काय आहे तर एक सिक्वेन्स, एक डी एन एची सिक्वेन्स, सपोज ह्युमनची डी एन ए सिक्वेन्स आहे, ती माऊसच्या डी एन ए सिक्वेन्ससोबत जर आपण कम्पेअर केली तर दोघांची डी एन ए सिक्वेन्स सारखी काही प्रमाणात असेलही, काही प्रमाण नसेलही. तर याचा अर्थ काय आहे की जो भाग डी एन एमध्ये वेगळा आहे आपल्या आणि माऊसच्या, तोच भाग किंवा तेच जीन माऊस असण्यासाठी त्याला किंवा माऊसच्या जे रेग्युलेशन आहेत त्याच्यासाठी रिस्पॉन्सिबल आहे. आणि दोज जीन्स विच आर डिफरंट फ्रॉम माऊस genome or DNA that are responsible specifically for human growth and development. So this will also give, याच्यापासून आपल्याला हे पण ठरवता येईल की कुठला ऑर्गॅनिझम कुठल्या ऑर्गॅनिझमपासून इव्हॉल्व झाला आहे किंवा उत्पन्न झाला आहे. सो the evolution process can be studied because the most similar sequences, सपोज आपण 10 सिक्वेन्स कंपेअर केले तर त्याच्यामध्ये 2 सिक्वेन्स खूप मॅच होत आहेत. इट मीन्स दॅट 2 ऑर्गॅनिझम्स आर ओरिजिनेटेड फ्रॉम अ वन कॉमन ancestor in which again we have to make this sequence analysis and we have to find its actual parent. सो जसं की आपलं ह्युमनमध्ये एक उदाहरण द्यायचं झालं तर मदर आणि फादरचे जे डी एन ए सिक्वेन्स आहे दॅट विल मोर लाईकली टू मॅच विथ दिअर पॅरेंट ऑर दिअर सन. सो लाईकवाईज you can find the evolutionary process. Then the from amino acid sequence there is a secondary structure of protein, then tertiary, then quaternary. So likewise from sequence to structure and from structure to function, relationship can be established with the help of this sequence analysis tool. Now there are some selected or very well known tools for primary sequence analysis. So first is BLAST. It is a search tool for DNA or protein sequence search based on identity. So how many nucleotides are identical in both sequence are studied with the help of BLAST. So BLAST is basically explained as basic local alignment search tool. Then next is HMMER, homologous protein sequence search from respective database. Then cluster omega will provide multiple sequence alignment, so more than 2 sequence can be analyzed in this cluster omega tool. Then Sequirome, so used for sequence profiling. Then Protparam, used to predict the physicochemical properties of protein. Then GISAW to find gene and to predict the splicing site in the selected DNA sequence. So then there is a Novo SNP, then ORF finder. So Novo SNP and ORF finder. Then PPP, virtual footprint, web GESTR, then gene scan, then software. These are the some well known and selected tools for sequence analysis. तर आता याचं वन बाय वन आपण स्टडी करणार आहोत. सो आय want to let you know that apart from this, the blast is very implicated in very different or in more these applications of bioinformatics in research, academics, and it is also run by NCBI, that is the National Center for Biotechnology Information. So that's why it is more having more accuracy, we can say, or it is user friendly to make analysis. So we will start with BLAST. So BLAST is basic local alignment search tool used generally for homology searching and similarity identification in the sequence of nucleotide and proteins. So this is a method used to execute fast homology searches between large sequence of biomolecules. The comparison usually carried out between the sequence as well as against sequence present in a biological database. So there are many developed version of BLAST, but NCBI BLAST is most popular. So there are separate version of such EBI BLAST, WU BLAST, also developed in order to increase the versatility and accuracy of algorithm. So the output obtained in each NCBI BLAST is given here in this video or in this picture. So on the basis of query sequence, different type of BLAST were available. In this there are different types of BLAST. That is, BLAST P compares amino acid sequence of 2 or more protein. Then BLAST N assess likeliness between 2 or more nucleotide sequence. Then BLAST TX, that is, तर आता ब्लास्ट पी मध्ये काय करणार, ब्लास्ट पी नावाप्रमाणेच ब्लास्ट पी कम्पेअर्स अमिनो ॲसिड सिक्वेन्स, म्हणजे दोन प्रोटीन सिक्वेन्सच्या जर दोन प्रोटीन सिक्वेन्स आपल्याला कम्पेअर करायचे असेल तर वी आर युजिंग ब्लास्ट पी, आणि दोन न्यूक्लियोटाइड सिक्वेन्स कम्पेअर करायचे असेल तर वी आर युजिंग ब्लास्ट एन. देन ब्लास्ट एक्स, that is used for align the translated nucleotide against a protein sequence database. So the translated nucleotides, so the nucleotides which is transcribed and then it is translated. So in this type of blast we are having a translated nucleotide which is compared with the protein sequence database. Then next type is T blast N. So search it search protein query against nucleotide sequence database. So the if you are giving protein sequence, then it is searching a nucleotide sequence from the database. Then another type is a T blast X. So it basically compares the translated nucleotide query against the translated nucleotide sequence database. So this is what we are saying T blast X. तर ट्रान्सलेटेड झालेली न्यूक्लियोटाइड सिक्वेन्स सोबत कंपेअर करणे. सो इन बोथ द केस क्युरी अँड सब्जेक्ट are translated nucleotide sequences. So this is what little bit about BLAST sequence analysis tool or sequence alignment tool. So the different versions of BLAST are there, that is PSI BLAST and FASTA, and it is basically a FASTA format used to give an input for a blast. So likewise there are HMMER, emboss, then cluster W, and then pattern hunter, then as we have above mentioned different types of tools. So gene scan and soft query. So these tools will most, or we can analyze if we understand these tools more accurately when we will do the practical for these tools. So up to now for this it is we conclude here for blast, and in next lecture we will again discuss with different types of tools, that is phylogenetic analysis tools. So thank you.